I spent past probably over 30 years studying cytotoxic T cells. Gosh, that's very long. And so mainly in virus infection, and most recently just started programming cancer. But really, for me, probably the infection, all this infection, because T cell has probably, I realize lots of T cell expert here, so probably there's no need to say T cell is extremely complicated, and can be good, but if it's not function properly, it can cause all sorts of problems. Instead of talking everything I've done, I thought I'd start from our work and probably focus on our work on SARS-CoV-2, because as a human T-cell immunologist, and we see, as Antonio probably agree, it's a very good opportunity, a human model to study T-cell and the natural history and the association with natural history of virus infection, primary virus infection, and from a T-cell priming to resolution and the long-term memory formation. So when the COVID started, our team had access to a very small cohort of primary infected patients, about 50. So our first question was, which part of the virus are frequently targeted by T cell, so-called immunodominance. And with the help from ProImmune, within 5 weeks since the pandemic hit the UK, we actually managed to identify and validate 6 very dominant T-cell responses. I saw your poster outside. That was the paper we published together with the ProImmune Tetramers and Pentamers. So that was a really great time. Not great time, I mean in the lab, very busy, very sad time. But in the lab, we've been working day and night, and with help with the ProImmune to providing us priceless Tetramers and Pentamers as a collaboration, but it was really fruitful collaboration. And since then, until now, we've been follow up this cohort to really try to understand some questions still not resolved, but we learned so much from COVID because the longitudinal cohort we have access to, and so at the moment we really focus on the factors that associate with the disease outcome, and doing a lot of omic type of study and using TCR as a link from the function to transcriptomic profiles and all that. I will give you a couple of examples. Also, what immune memory like 4 years after infection, and the implication and cross-reactivity and implication for probably future infection. So this is all started. So this is the key team in the lab, and really at the beginning of COVID to really, sorry, I should put it ProImmune and Jeremy here. But all these tetramers are made by ProImmune. So we identify these immunodominant epitopes and still working on that. So I'm going to give you a few examples what we are doing now on this very dominant, where is the, oh, okay, it's here. So this is the B702 MP restricted. MP is a very dominant antigen we are really interested in. Also the spike CD4 responses, again very dominant. so this is a kind of pipeline, research pipeline we established during the COVID time. So we have a longitudinal sample since the convalescence until I think it's more than so. So now we bleed the donors every 1 year, and just to see what immuno profile like. And so when we get the PBMC, we sort with the tetramer or cytokine profile, depends on the frequency. We have a different method to enrich the tetramer positive or pentamer positive cells. Then we use half of the cells for T cell clone and expansion, half for single cell sequencing, because the tetramer sorted cells from PBMC, as you know, is very not a lot. Normally, sometimes you get 100 or 200, so you can't do 10x. So we use SmartSeq, and which is quite technically it's actually quite challenging. But we kind of managed, seems like. And then for the T cell clone, we do T cell receptor profile and do all sorts of functional assay, and we actually also, for some very interesting clone pairs, we do proteomic analysis as well. Then we analyze T cell receptor repertoire and match the single cell sequencing with the antigen specificity and functionality. So this is kind of become a pipeline, the lab, what we do. But at the moment, we kind of, the focus is beyond the phenotype now, so we really try to work out, because from the T cell receptor repertoire analysis, it's become very interesting now. We observe lots of public T cell receptors in COVID, but they have very different pattern. It's epitope dependent, and some have a very dominant beta, and some have very dominant alpha, but pairing with either private or public beta or alpha. So they have, so we find some really interesting patterns. So we focus on try to understand where this public T cell receptor come from and what they mean for the future infection, as well as cross-reactivity. So, so actually, just step back about why I'm interested in public T cell receptor is actually one of my postdoc projects led me to really get interested in the T cell receptor. It all started from our HIV work, and we see there's a very strong association with the public T cell receptor with the long-term non-progressor. So it's HIV positive patient, but they don't get don't get sick, so they have very low viral load. So what we found is they all use same V beta, but not other. So later we did structure for the T cell receptor. That time we can't do the complex. What we found, so two things we found. One thing is this public T cell receptor seems like can accommodate lots of variants, so they're very highly cross-reactive. Why the private TCR not? Another thing is this TCR is exclusively using very long CDR3 loop. So so CDR3 is critical for peptide recognition. So, and it's quite wobbly. So we hypothesized that time, because this long loop CDR3 gave them the flexibility to accommodate more variant. So when we observed, the first public T cell receptor we observed is this very dominant B7 restricted MP epitope. So as you can see here, and B7, so among the B7 donors, we have 19 in our cohort. Almost 80% of them make this response, so it's extremely dominant. And it seems like associated with a mild symptom. So when we look at the T cell receptor, so there's so in the mild patients, it's like large expansion of public T cell receptor frequency than the severe cases. And when we look at this public, at that time, everybody believed, because the sequence of this peptide is identical from the seasonal coronavirus, so people assuming this is a pre-existing T cells from the memory from the previous coronavirus infection. But in fact, when we look at the phenotype of the, because we did single cell naive phenotype, at that time, actually, Australia group also published, actually published before us, basically saying the same thing. So we believe this is a high-frequency precursor rather than pre-existing memory. So we published this data, and so we moved on. At the moment, we're thinking about doing the structure analysis and some more analysis, but I think we moved on to the second epitope. A second group of epitope is a spike-specific CD4. While we also, so this is, again, is very dominant, and it's two of them are DR1501 restricted, and one is DP0401. DP0401 is half of the population have this allele. I think then, so again, the O3 epitope is very, very dominant. So when we look at the T-cell receptor repertoire exclusively, each of them using a very, very dominant, just one. Among all the, most of our donors we look at, is they use universal alphas, which is quite unusual and surprising because most, in many cases, you see public T-cell receptor, but they're not dominant. But in this case, they're very dominant. However, the paired betas, they're very diverse. So in this case, and we do see public pair for alpha and beta, but the main thing is the public alphas for all three. And we actually did look at other like internal CD4s. Looks like this is really specific for these three dominant. But it could be a reason because they're so dominant, they probably are selected during the acute infection. We don't know. It's something we want to find out. And we follow this for 4 years. You can see this dominant. So the blue one is public T-cell receptor. It's still dominant over the 4 years and time when we follow up. So they maintained. And while the privately changed the profiles. And then, so let's become really interesting because our cohort is too small to look at clinical association properly. So there's two very large cohort in the database. One is Merkov cohort and the other Batcher's cohort. So laid down the bulk TCR sequencing with the alpha and the beta data available. So we look at the association with the clinical outcome. Both cohorts shows this public T-cell receptor we identified is associated with mild disease, which is really interesting. Then we look at there's another cohort, pre-COVID cohort, also have a T-cell receptor sequencing. What we see, again, is similar as what we see in the B7, is the precursor frequency for this TCR is really high in healthy donors. So again, we think the reason we have this is the precursor frequency, high precursor frequency of these alphas for some reason, rather than pre-existing immunity to coronavirus. So we look at, the CD4 T cells, when the high avidity T cells and also like highly differentiated T cells, normally they have accumulated cytotoxicity signaling. So then we look at the cytotoxicity of this spike CD4 response. What we see is over the 4 years, there's accumulation of cytotoxicity in this cohort. So the CD4 cells become more cytotoxic after 4 years infection. This could be due to the multiple antigen exposure, we think, either due to the infection or the vaccination, because this is spike. And we are currently actually comparing the internal protein phenotype, and which I think is very distinct. And we also compare the cytotoxicity with the virus suppression assay, while we're using the co-cultured T-cell clones with ACE2 transduced target cell lines. It's correlates really well with cytotoxicity. So it's functional relevant. And then when we look at the molecules that contribute to the CD4, as we know, the granzyme B is the main, apart from perforin, is the main cytotoxin contributor to killing. But in this case, CD4 is granzyme A, which is also reported by somebody else, I think, last year. But anyway, so at least the cytotoxicity of this set of T cells is contributed by granzyme A, as we show here. Yeah, so this is the T cell lines without can kill and not kill. So we look at we did a proteomic analysis and of this and RNA-seq transcriptomic profile. Both shows the same thing. And is increased, granzyme A again increased over the 4 years' time, and validate what we see, the increased cytotoxic and killing in the previous study. So this paper now, I think, is impressed. I think finally the reviewer is happy, is impressed in the Nature Communication. If you're interested, we uploaded all the raw data. So if anybody wants to look at the transcriptomic data, T-cell receptor data, so it's all there. And if you have any interest, just let us know and want more data or want clarity. So move on from that, and we also look at there's another responses from the early findings, this ORF3A0101. And instead of protection, and this looks like this response, again, is very dominant. Almost all the patients we look at make this response at that time. And it's really associated with the disease severity. And at that time, we have a hypothesis of probably ORF-A is quite late protein, which is quite different from Antonia and Nina's, the SP, the early protein. So I think they're late, but they have very high avidity, so they probably contributed to the pathogenesis rather than the, we don't know, but that's our guess. So when we look at the T-cell receptor repertoire, again, we found public and private T-cell receptor usage. And it's a very dominant T-cell receptor here. It's TCRR2, I think. It's, but the private is all very diverse. So we... So this one, because it's become quite clear to us, there's something about the public T-cell receptor and the cross-reactivity with all the functional data. I didn't have time to show all of you the cross-reactivity to the natural variant and all the potential variant. But it feels like there's probably a link between the public T-cell receptor and the cross-reactivity. And there might be a structural explanation for that. So we moved on to collaborate with the biochemistry and a structural biologist in Oxford. So Ricardo's group is he's an immunobiologist, I would say, and so he studied biochemistry, but then later on moved to the Chris Gashia's lab, really to looking into the T-cell receptor cross-reactivity and antigen discovery through the East Library system. So when we recruit him, he brought the whole system back so we can look into the T-cell receptor cross-reactivity properly using his East Library. But in this case, so first we kind of compared the T-cell clones bearing public and private T-cell receptor. So they all show very similar avidity. So this is EC50 to measure the avidity of T cell response to when they meet the antigen-presenting cells. So it's very similar. Then Ricardo make the alpha-beta complex protein. It's actually quite tricky to make it. And he made it, and so as you can see, it's expressed very well, the heterodimer. And then he did this Berkel assay to measure the affinity properly. So again, shows similar, but it's relatively, William probably know, the affinity actually is not that high, because for crystal structure, you need a quite high affinity peptide, right? So the KD, this is a macromolar level. So I think it's similar, but not identical. Then we moved on to Peijun. Peijun is our cryo-EM queen. So she's really the world leader for the cryo-EM. So, but she has no clue about what we're talking about. When she asked us the T cell how big it is, it's tiny. But she actually, with her team, tried. Ricardo produced this beautiful protein, enable her to try the cryo-EM. They play with the system, the method. At the end, after a year, they made it work. So they make actually quite good resolution, 3 Armstrong. So for both public and the private T cell receptor. So as you can see, this is public and the private. For the public receptor resolution is a little bit low, and the structure for the side chain, the beta, and the MHC peptide contact area is okay. But the beta-2M interaction with beta-2M is too wobbly, according to them. I'm not structural biologist. This is what I understand. So they can't really see it properly. But for the private, it's much better. So they think it's probably likely due to the flexibility. But it's probably contribute to flexibility of this public T cell receptor. That's why they can accommodate more variant. So that's what they think when they. This is the immediate thinking. Then when they look into the details, because although you can't see the beta-2M, but the resolution is high enough for them modeling the side chains, most of the side chains. So they can see what they notice is the key message from this. I'm not going to detail to each amino acid interaction. The conformation of the MHC peptide and their interface were nearly identical between the two complexes. However, the TCR CDR3 loop engaged the antigen peptide more extensively in the private than the public. So it's more like a verified what they think at the beginning. Then when they look into the interaction profiles, and so it's, although it's a very similar overall architecture, but the CDR's interaction profile is different markedly. And so this is the pink, I think, is the public. Blue is the private. So then they look into the really details. Anyway, this I think is too much for me to understand the details. But but the key information is the CDR3 loop of the TCR public receptor is positioned much closer. So they're very much closer contact, and the private it seems like establishing more extensive contact with the alpha helix, and the public receptor interaction are primarily concentrated on the alpha-2, as you can see here, but private seems like distribute more evenly. So this differentially interaction pattern again suggests the increased stability of the private peptide MHC complex, as well as the flexibility and of this T cell receptor. Again, suggests public T cell receptor is much more flexible than and private. Then they finally they kind of used there's not much T cell receptor structure in complex with Pentamers C in the literature, so they just piled up everything, and to use that as a reference database to compare our TCR public and private. So the conclusion is the public T cell receptor distinguished the T cell private by the CDR engagement mode, and so allowed to accommodate a broader range of for the public T cell receptor. So this flexibility, engagement with the CDRs enable them to accommodate more epitope variant. So that's kind of our conclusion for this part of the study. And so now we have a long list of the public and the private T cell receptor paired against the same epitope restricted by same HLA. So we try to see if we can work out the rules which might govern the different recognition of the peptide and their variant. So this is kind of ongoing work, and the paper is at the moment is under review, and. So move on. So the list is, so we are doing a lot of public T cell receptor work at the moment. And moving on from that, I just want to, how long I have left? 5 minutes, great. So very quickly, it's relevant with that. We also, because we were, during the COVID time, when we evaluate, I got a little bit increased anxiety about using overlapping peptide as a standard thing to evaluate T cell response. So we actually spent lots of time reinvent the old-fashioned T cell assay using vaccinia virus to make the wild type virus as well as the wild type spike as well as the variant into the vaccinia virus. In that case, we can measure the T cell response to naturally processed and presented epitope. And not only consider the variant within the epitope, we also consider the variant within the flanking region. So we covered escape from both within the epitope as well as the mutations might affect antigen processing and presentation. So this is the collaboration with Jeff Smith, who is the world-leading pox virus expert. So he made all these nice vaccinia virus. So we kind of established that system and to evaluate the T cell response on top of the overlapping peptide. Because the work is published, I'm not going to. So this system is actually brilliant. We can really evaluate at the population level the escape. And through this system, we were able to identify a few escape mutations within the flanking region. So we use the same system because I'm part of the UKRI monkeypox consortium, and so responsible for the T cell evaluation. So we have access to the two study cohort. One is the convalescent samples from UK monkeypox infected patient, and also in Oxford there were ongoing clinical trials of MVA vaccine. So we took the sample from MVA, similar time point from the vaccine cohort as a control for the convalescent sample, which we take the blood between 2 to 4 months. So we compare the response between the two study cohort, and using the vaccinia virus and as infected target cells as the main antigen. So what we see is we see a very good CD4 and CD8 response in monkeypox infected individual, and we also identify 7 dominant monkeypox specific T cell response, and with one of these almost 100% donors make this response. So we moved on to did single cell analysis to look at the transcriptomic profile for this. And so cut the story short, and so we see much larger clonal expansion in infected than vaccinated. As you can see here, this is the paired alpha and beta. So large clonal expansion, some public, we haven't really looked into it. So this is the UMAP and alpha and beta. And so the pink is convalescent and blue is vaccinated alphas. So as you can see, the convalescent has much more clonal expanded population in both. And when we look at the transcriptomic profile and in infected cohort, they're much more activated and show much greater cytotoxicity, and as you can see here. And yeah, so basically, so now we end up lots of TCRs that shows good and bad cross-reactivity. So the lab actually spent a lot of time to try to dissect if there's any structure determines in terms of cross-reactivity, publicity, and also the precursors, where they come from, and especially the tissue relevance. Yeah. So lots of actually people involved since the COVID started, and so the people highlighted the ones contribute to work I just presented. Thank you. Oh, this is the, oh.
Hello.
This is the lab.
So you showed at the beginning that the T cells with the T cells with the public TCR,
yeah,
they were all naive in the non-infected.
Just the two. We only I don't know if the rest, but we had looked at the B7, CD4, the O3, CD4 spike, and A3 in the public database is all naive. Oh no, actually, it's a pre-COVID. So. Yes. We only checked the transcriptomic profile for the B7. We didn't check the other two, but we found the TCR pre-COVID.
And is this region that this is targeting really different among different coronaviruses?
Yeah, so apart from B7, it's identical. A3 and the old spike is completely different. So it's all the sequencing. It's completely different from the
yeah, because corona. Otherwise, if they were.
But I didn't include the one you and in your paper. I think that we only look at the coronavirus, like whatever the most common. I think you have a very different one we missed in your paper. Yeah, maybe that one. Yeah.
No, I was just thinking because you said on one hand these public TCRs are more flexible.
Yeah.
And allow more mutations. So in a way, we would expect them to be more prone to be cross-reactive than non-public T cells with non-public TCRs. No.
This, I don't know because this is very little data, and it could be very. And I think that's why I think we just need to see more natural variant. It's a very good question. I don't know. It's one of the things we don't know. Yeah.
Yeah, great talk. So in your talk, you mentioned about cytotoxic CD4 T cell, the ingredient A, the dependency. Then how do they recognize cytotoxic target cell by TCR image class 2 or other NK-like receptor? Or?
No, for this one, because they have all these cytotoxic molecules, so it's direct killing.
But for the in front of the target cell, they need to be activated by TCR. No?
Yes, it's through TCR.
So my question is the target cell, do they express class 2 MHC?
Yes, so it's all matched. The killing only through the matched class 2.
Okay, yeah.
So without matching, it's not non-specific killing. It's a specific killing.
Okay, thank you, Professor Dong, about the excellent presentation. My question is about, you showed the high collaboration between the public TCR and the mild infection, right?
Yeah.
Is it means the vaccine based on this peptide can be a great vaccine for the?
Yes, I think so. The dominant, but that's what I hope to say. I'm not. Yeah, they're dominant if you come. I think for CD8 protection, I think the early the better. If it's late, it can be a problem. I think we believe that could be the problem for the ORF3A. I don't know. But the early ones, I think Nina and Antonia already demonstrate you need very early response to. So that's why we think B7 is protective because they have high frequency precursors. And so as CD4 spike, those ones probably are good because they have high frequency precursor.
Okay, thank you.
So just to clarify, so these are CD4s, but recognizing MHC class 1, is that?
Class 2.
But if it's class 2, it's going to be highly restricted to immune cells. So I'm just trying to understand how it's mediating the antiviral effect. If only a very restricted number of cells express MHC class 2, then the cells infected by the virus are not going to be expressing MHC class 2.
That's a very good question. I don't know, but it's this T cell. Honest, I don't know the CD4. These are definitely killers.
Yeah.
But if you compare these killers, the killing activity with CD8 is much less. So honestly, I don't know if it's in the real situation. Like you said, it depends on the class 2 expression. They can be an effector or most timely because the CD4 killing is driven by the differentiation and high avidity. And the method we use could be artificial, I don't know. But they do have a content much higher cytotoxicity. So in the specific environment, when the cells are activated, the class 2 is upregulated, that can be a problem if this CD4 start killing because this is not their function. But in the microenvironment, in the inflamed condition, that's why we're very keen to, at the moment, we have a 30 post-mortem paraffin-embedded lung tissues. So that's exactly the thing we want to try to link the clonal expansive TCRs in tissue to see if in the disease area and the non-disease area, if there is accumulation of these cells, especially public T cell receptors, and to see if they actually contribute direct. And then we can do the standing on the class 2 as well. But this is definitely a really good question. But you're right, in the normal condition, class 2 probably low, but in extreme condition, everything is perturbed, then it's become a problem.
I mean, the other possibility is that these CD4s are actually there to protect against viral infection of other immune cells, such as the monocytes and the alveolar macrophages. And by controlling the virus in those cells, maybe that's having an overall impact on the infection, if the virus is replicating or at least infecting those cells.
Yeah, in COVID case, it's relevant to lung COVID because now we have actually tissue and also gut tissue COVID patient. It will be really interesting to look at other side as well. I don't know the answer, but it's just the good thing is because this TCR is so dominant, and actually it's like the DP0401, half the population have it. That means without needing to study T cells, for people have the access to the tissue and omic data, they can pin down this T-cell receptor to say, okay, I know, because they are shared the publicity from the already published data, then I know they are cytotoxic against this epitope, then they can interpret data. I think that's our contribution, I guess, because they are dominant alphas.
Thank you.